Hey everyone, it's Chris, the host of Is It Just Speculation? What if I told you that you can make your very own podcast for free and you can make money from your podcast with no minimum listenership required? That's Anchor. If you haven't heard about Anchor, it's the easiest way to make a podcast, and that's what I use. It has all the tools that you need in one place so you can record and edit your podcast right from your phone or computer. So you can work on the go or you can work at home in the comfort of your PJs. That's what I do sometimes. Don't tell nobody though. Anchor also distributes your podcast for you so it can be heard on Spotify, Apple Podcasts, and many more places all around the world within a matter of moments. That's the beauty of Anchor. It really lets you have a message and get it out there. You have your own voice, guys. It's everything that you need to make a podcast in one convenient place. Please download the free Anchor app or go to anchor.fm to get started. You're missing out if you don't. Hey, welcome to the podcast. This is Chris, and it's just speculation. Stick around for more. I'm excited to be joined by our special guest today, Dr. Azza Gadir. She's an immunologist. She completed her postdoctoral training at Harvard Medical School, Boston's Children's Hospital, you might have heard of it, where her published research was focused on the immune system, food allergies, and the gut microbiome. Azza grew up in London and holds a PhD in immunology from University College London, a master's degree in the immunology of infectious diseases from the London School of Hygiene and Tropical Medicine, and an undergraduate degree in biochemistry from King's College London, all in the UK. All right, so Azza, is that correct?
Yes, that's correct.
Okay. All right. If you can start by telling us about yourself, your background, and as far as your work and education and what you do.
Sure. So I'm an immunologist by training. So I have a master's in the immunology of infectious diseases from the London School of Hygiene and Tropical Medicine. And I also have a PhD in immunology. My project was mainly focused on lupus disease and understanding the immune system and why it can drive lupus. And then after that, I actually moved to Boston to do my postdoctoral fellowship at Harvard Med School and Boston Children's Hospital, where I was focused on, again, the immune system, but particularly looking at food allergies and how the immune system causes food allergies and the role of the microbiome in that. So after that, I actually moved over into industry. I left academia and I joined a startup working on probiotics for about 2 years. And then as of March of this year, I work for a cancer immunology startup. And so my job is to kind of look at the science and try and kind of design treatments around the research as it comes out.
That's great. So, your background is a big reason why I wanted to reach out to you, just to get some information out for people that maybe are not as well versed in science or maybe the studies, and they don't really take the time to look at that. And, you know, even if they did, they probably wouldn't understand all of it. So one of the big things that is an issue nowadays is misinformation. How do you combat misinformation, and particularly with preprints? How can you manage taking preprints that might contradict each other?
Yeah, it's a little bit hard. I think with misinformation in general, there's a lot of it. And I think that especially with COVID, the information has been coming out very quickly, and it's hard to kind of, as you said, discern between what's real information and what isn't. I think the main thing to remember is that scientific publications, they were coming during COVID, they were coming out, the science was moving faster than the peer review, right? Because when you design a study and you write it up and you get results, you have to go through the peer review process, which is when it gets sent out to other experts that you don't know in the field anonymously, and then they critique it and it comes back to you. And so that process takes a little bit of time because you need human beings to make the time to read. So that's why it's important to remember that because the science was coming out so fast, you're right that we were getting a lot of preprints that hadn't gone through the peer review process yet. So it was hard for most of us to see immediately if there were mistakes in the paper. But I think the important thing is to remember that science is a consensus. So it's lots of different like papers, it's not just one paper changes everything. It's usually a bunch of papers that build on each other or build on a message. And then you look at the consensus and then you make a decision. And so generally, again, like that's why there's a saying where like science doesn't worship a person. It's generally consensus. And so I would say that when it comes to preprints, to read them, try and gather information from them, but just keep an open mind that the consensus may be different and to kind of hold off on strong judgment. I hope that made sense.
Yes, that's pretty great. That definitely helps understand kind of what to do with those massive amounts of studies coming in. And I think a lot of that might just be because it's such a big issue. There's a lot of funding going towards the studies. And so maybe that just encourages more and more. Is it hard to do a study in a particular area of the world and then apply that same information to maybe a different place where maybe there's better healthcare options or maybe just a complete different living situation as far as maybe not as worse off as developing countries?
Yeah, that's potentially true, which is why you have to remember these things when you're analyzing the paper. And I think generally when I know that like the word expert gets thrown around a lot, but I think experts are people that have spent a substantial amount of their time. It's not necessarily credentials, but it's people that have spent a lot of time researching a specific topic, so can understand context and will be able to see through mistakes like that in studies. And so in answer to your first question, yes, studies can be really hard to do. It depends on the type of study, right? Because you have cell studies, cell culture studies. You also have animal studies, which can move a little bit slower. And then you have clinical trials where you're looking at the general population or doing observational trials to see what's going on. And so generally these things require time. So yeah, they are difficult to do. You always have to remember, you have to make sure that you have the right control groups in order to interpret the research. So you have to make sure that like, you don't just have a group that gets the vaccine, for example, you also need a group that gets the placebo and that they're blinded properly, and like all of this, everything is done the right way so that like you can't then have the data misinterpreted. And so yeah, I in answer to that question, yes, they are really, really hard to do. Yep.
Yeah, and that's part of the peer review process where they basically go in and replicate the studies if they can, maybe even poke holes in maybe the methods. Is that something that usually dismisses a lot of preprint studies, or for the most part, are a lot of the studies usually pretty well off to where they still can be replicated?
It depends, right? Because in terms of replication, that generally comes with as other groups publish, then they corroborate. So for example, you'll have like some pretty big immunology labs in many different countries that are like have access to good patient cohorts and have a history of publishing research that makes it through the peer review process really well. And so I think that at the beginning of the pandemic, it was probably way harder, that there was a lot of junk that was coming out because, it was a brand new virus. Everybody was focused on it. It was really infectious. And there was like a lot of research that was being done on it. And so at the beginning of the pandemic, that was the case. Whereas I think at this point, we're moving slowly towards, like you can tell from the authors on the paper if they're gonna, if they have the right access again to the patients, or if they have a history of analyzing data properly. And so like those are the types of things I think and patterns that people will start to look for. But don't forget that preprints will eventually make it into publication, so you'll know. So if you see a preprint and you wait a little bit, eventually, if it's a good paper, it will pass the peer review process, and then you'll hear that it got published in a journal, and then that means that it's passed. Whereas the preprints that never published, I think it kind of says a lot about perhaps the quality of the work.
Yeah, you actually reminded me of a point. So looking at the authors who do those studies, who put their names on there, and you can kind of track their history as far as how right they are or how wrong they are.
Yeah.
A lot of the, probably not misinformation, but a lot of the confusion would be people's individual doctors. Because if you have a doctor title and you have your own practice, no matter what your recommendation is going to be, most likely the patient's going to take that, especially if you've had the same doctor your whole life. Maybe your mom had the same doctor, if that's the case.
If there's trust there. Yep.
Yeah. So how do you, would you do the same thing and kind of look at that doctor's history and maybe see what they've done in the past that may have not turned out to be true? And could you really do that? Because there's not a lot of times where you can probably go back and trace that one doctor to maybe make a big decision, like during the pandemic, whether you
Yeah.
give that.
So yeah, totally. It's not easy. I agree. It's not easy to know, and it's not always information that's easy to access. But I would say that if your doctor recommends something, I mean, generally, as you said, people have trust with their doctors and they have a history. But if you would like to corroborate what your doctor's saying, ideally you would go to some of the bigger boards. Like, for example, kind of like what the general consensus is or what the government's advising. You can go and find that on the government website. And generally government guidance comes from consensus. So it's what the scientific community at the time has known as the consensus at that point. And so I think that's a way, if you then see that your doctor's saying something that is different from what the majority of people are recommending, then I would ask them, can you provide me evidence or data? I would really like to understand why your opinion is so different. You know, so I think that I agree with you, it's not easy, but these are some of the small ways that can perhaps help people make those decisions.
Yeah, and to that point, we recently did see the consensus play out where boosters were not recommended for everybody, and it was just actually given to people at high risk or older populations. So even, you know, real world, you can see that some people in the agency and the FDA, CDC may have been for booster shots for everybody, and then it ended up
Yeah.
being recommended against, at least right now. If you want, can you give me some thoughts on that? Maybe why that was the case?
Yeah, sure. So I think that this is actually a really complicated issue, like in terms of boosters. So if you don't mind, can I just explain quickly, just a very, like for anyone listening, what a vaccine actually is and why
Yes,
it differs?
please.
And yeah, so I just want to explain one thing, which is your immune system, right? It gets bombarded. Things go in through your nose, your eyes, through different orifices in your body, like lots of times in a day, right? From the minute that you're born. And your immune system is generally good at saying this thing should not be in the body, it needs to get out. And so the main thing to remember is that you have your innate immune system, which is the beginning part, which is if something, you know, you breathe something in or whatever, you have these cells that patrol the body and they're able to say, this thing shouldn't be in the body, I need to get it out. And they'll punch to get it out, right? But what happens is that on occasion, and actually quite rarely, your innate immune system encounters something that overwhelms it. So what happens, for example, with SARS-CoV-2 is that your innate immune system sees it, it's punching for a couple of days, it can't get it out, and it realizes. So at one point, it grabs a chunk of the thing, and then it runs to your lymph nodes where you have your B cells and your T cells, and it says to them, this thing is in the body, I can't get it out, can you guys start training? And so what happens is that you have the second part of your immune system that where your B cells and your T cells are part of, that is specific. They start to make more of themselves that are specific to that thing that's being presented to them. And then after a few days, they rush out and your antibodies will cling on to any bits of virus that's floating around so that your immune system can chomp it away. And your T cells will go and basically kill any cell that's infected. And it does that. It's a very specific process. So the best thing about activating these cells is that not only are they specific, but they actually come with memory. So even after the virus is cleared, they can go and hide in your system. And then if you're ever exposed to the thing again, it rushes out and it gets rid of it for you. So that's what's called your memory immune system. And so because your memory immune system or that compartment is so specific, you can show it anything that you want, but if it doesn't see the thing itself, it's not going to remember it. And so vaccines are essentially a way of showing your immune system something or a part of like a virus in the case of these vaccines, so that if your immune system can build these like cells and keep it in their back pocket, and so that if it ever encounters the virus, they can rush out immediately to protect you and prevent, right? And so that's just generally how it works. And then with vaccines, you have a prime and a boost. You tend to have two shots. I know the J&J was only one, but the others were a prime and a boost. And the first shot is when your immune system goes through that process where it generates those memory immune cells, right? And then the boost is basically when you get exposed again after a few weeks, your immune system does this thing where it goes, hold on a second, why am I seeing this thing again? I thought I just fought it off. And so what it does is that it goes in amongst the pool of B cells and T cells that you've made and selects like the best players. And so that's why your immune system actually gets better over time at identifying the thing. And so the COVID vaccines that we have currently authorized in the US only have one part of the virus. The virus has 29 proteins. These vaccines only have one, and it's the spike protein. So they're all different ways of showing your immune system the spike protein so that you can go and make those spike specific B cells and spike specific T cells, and they can stay in your back pocket. And if you see the virus that has spike on its surface, you have those cells ready to go. And so when we talk about boosters, right, it's not unsafe. Your immune system, again, like even with the vaccine, you can go and be exposed to COVID, and you can get like your memory immune responses can keep getting stimulated.
Yeah.
So we know that boosters aren't unsafe, right, conceptually, and what we know about the immune system and knowing what we know about other vaccine schedules and the other ways that other infectious diseases work. We know that like repeat exposure to your immune system isn't going to cause, like, isn't going to be unsafe, right? But the thing, that's why the conversation from a theoretical perspective, like when people say, you know, get a booster if you want to get a booster, or, you know, or don't get a booster, it's that it's not unsafe, but the question is whether it works and whether we should be like in the developed world kind of giving out these vaccines willy-nilly as boosters when there's still so much of the world that doesn't have access. So I think that that's the question. It's like a way up of a situation. And so when the FDA met, they looked at the data to say, do we have data that shows that at this point, in the majority of people who are not high risk or over, I think, the age of, I think it's 64. Don't quote me on that. I need to double check, but I think it's like the elderly and also people who are immunocompromised. Those are people that we see their immune responses maybe going down and they're getting sicker, right, as time goes by. Right. So we should just give their immune system a quick flash and a boost again so it can generate those cells for them. But they looked at the data in people who are under that age group, and they found that there wasn't sufficient data to say that it was something that was needed, that actually all of the data supports that if you've had a full vaccine at this point, that you shouldn't have a problem if you're exposed in terms of hospitalization or death, in terms of if you're exposed to the virus. And so the FDA met to kind of debate the risks and like whether it's worth giving people a booster just in case, or whether it's worth, do we have data to show it whether it's needed? And the science so far, because we're still waiting, we're still monitoring these responses over time, doesn't support that the majority of us need a booster at this time. And so that's why they made that decision. And I think, you know, a lot of people disagree with that decision. I mean, I know some major immunologists that are like, if we have a supply, we should just start handing it out anyway, right. Just in case to be, you know, like this is an infectious virus that's right. Just,
why wait
like,
for it to get bad again, basically.
Exactly. So that's more the question. It's not, I don't think it's a black and white answer, if that makes sense. And I personally can't say that I know exactly where I stand. I think I support the FDA's decision because we don't have that data yet, and we can't just make decisions without data. Right. But in terms of like, yeah, I think it's a difficult question. Yep.
Well, thank you for touching on that. I think that kind of leads into my next question. And, you know, why is it that we're seeing the efficacy rate drop down after around 8 months? And do you know kind of what the protection rate would be? I know with the vaccine is probably like in the 90% for protection, if, you know, depending on what vaccine it is. Do we know kind of what the rate is after that 8 months?
Yeah, so I think that this again can be a difficult question to answer because there's so much that's involved. Mm-hmm. So first of all, the first thing that I want to, that's kind of been, I guess, a bit of a been misinterpreted often, is that people will look at, so some studies will look at things like antibody responses going down, okay? Right. And they'll say, over time, people are seeing reduced levels of antibodies in their system, which means that the efficacy of the viruses of the vaccine is going down. However, that doesn't completely hold water because something, you know, when I was saying to you that your cells go and hide away until you're exposed again and then they come out. Yes. That's something that's really cool actually that the immune system does, is that when it realizes the coast is clear, after a while, like after weeks, your B cells, they won't just continue chugging out antibodies, right? Like it just doesn't do that. What happens is that the cells that make the antibodies stop, and then the cells go and hide away. So that means that like antibodies waning over time is something that's expected with infectious disease in general. The only way you truly know whether somebody had protection that went down is if they were exposed again and their antibody levels were weaker than the first time. right? And so
Yeah.
I just want to say that like a lot of the studies misinterpreted antibody, they misinterpreted that, and they were like, antibodies waning over time, it means the vaccine efficacy is going down. But in the clinical trials, we looked at the efficacy of the vaccines at relieving hospitals, right? So that's stopping people from dying, which they did, and preventing severe disease, which they're still continuing to do. We're still seeing that in the emergency rooms, the vast majority of people that are still in the ER for COVID are people who are unvaccinated. So the vaccines are continuing to work. I think that the issue, the reason why I said it was complicated is because the answer to this depends on the variants. And variants basically, because we have such uncontrolled spread in so much of the world, right, including here in the US, where people aren't social distancing or masking, but they're also not vaccinating. So this virus is running rampant. And viruses, you know, like they hijack your own cells and your own cells' machinery in order to make more of themselves. And so the more time you buy them to hang around and linger in human cells, the more likely it is that they're going to mutate. And the majority of their mutations don't mean anything. You know, like every time a virus divides, it mutates. And very rarely do those mutations result in the virus behaving differently. But I think that the main thing here is that the more time we buy them, the more likely it is that we're going to see newer variants. And that's why currently we're averaging a new variant every couple of months. And I think that while it's still, you know, so uncontrolled, even though the vaccines are helping, and we can see that in like states and in areas where there's a high vaccine rate, that the cases are lower, and also the hospitals have less people in them. So we know that the vaccines are working, but it's just one of those questions where it's like time will tell, because we may eventually be able to get a new variant that will be a little bit better at evading a little bit of the vaccine. But like in general, the vaccines are continuing to work, and they are neutralizing against every single variant that's been identified to date.
Yeah, I think you made a really good point, because one of the studies that I was actually looking at was saying that there was, for the vaccine portion of the group, there was a 40% antibody decrease per month versus with the, you know, somebody who recovered from COVID naturally, with a 5% decrease. So that was one of those studies that showed me like, oh, like maybe the natural immunity might be longer lasting, or maybe not better because you have to get sick to get it, but maybe longer lasting. So for you to make that point that, you know, your body's not always going to be producing those antibodies if it doesn't need to, because that's just a waste of energy for the body.
Yeah.
I think that definitely hits a point that a lot of people miss, especially with those kind of studies.
And also, like, that's a very good point in terms of the study that you said you've read. It would, so we know that, again, like the vaccines have spike protein in them, the virus has spike plus the other, you know, proteins. And so we know that when you get naturally infected, the virus persists, right? It's in your system for like a little while. It's like a few days, potentially longer. I mean, there are some people who test positive for a couple of months after, and that's associated with long COVID. But the virus persists in your system for a time, which is why it's actually associated with so many long-term effects. Whereas the vaccines themselves are like, they're in such low doses, right? They're in an optimized dose from the trials. That means that in the majority of us, our immune system will respond, and our innate immune responses will be overwhelmed enough that it'll go and activate those adaptive. And so to your point, if you saw persistent antibody responses in people who got infected naturally, that would correlate with the fact that the virus is in their system for longer, whereas these vaccines don't last in your system for longer than a few days. And that's it. Like they last in your system for a way less amount of time than the virus, with, you know, replicating and mutating.
Yeah. Yeah, that's a great point. And you mentioned long COVID. That's one thing I wanted to ask you about, because I don't know if a lot of people really know about long COVID or what it is. Can you talk a little bit about that and what we know so far?
Yeah, so long COVID is basically that in a number of people who got infected with the SARS-CoV-2 virus, they were continuing to see symptoms for like months and months, even after their primary infection. In some of those people, if you test them, they're actually PCR negative. So they don't have any persistent virus in their system, but they still have symptoms that are persisting through. And I think that's one of the main things that people have to remember about this virus that caused the panic, that we've seen, is that
Right.
not only is the virus, really infectious, and that the disease itself is unpredictable, like we've seen healthy people and unhealthy people get wiped out by it. But I think that the main thing is also that there's this long COVID thing, which is that up to 30% of people, right now we're still measuring the data, but up to 30% of people maintain a symptom long term, even after they clear the virus completely. And so it looks like it might be something autoimmune related, or like a chronic, kind of autoimmune like syndrome, but we're still kind of trying to understand more about it. And I think that it's really important that there is a substantial number of these people, and these aren't people who are high risk. It's like young, healthy people, right? A lot of them are basically, some of them, their quality of life is so reduced that they're finding it difficult to go back to work. They're unable to get out of bed. It just depends on the symptom. And so it's one of the main things is that the vaccines are effective at preventing infection and transmission, at reducing infection and transmission, and they're effective at reducing the incidences of long COVID. There is a small percentage of people who are reporting long COVID even with the vaccine, but that's because, again, you have to remember that your immune system can get a head start on this virus. But if you put yourself in a situation, or if you are exposed in a situation out of your control where there's a lot of viral load in your environment, that will be able to overwhelm those immune responses. But still, it'll get a head start and it'll eventually win. It's just, it might be a battle. And in a small number of people that are vaccinated, they are getting long COVID.
So is there a chance that, after you're exposed, maybe you didn't get the vaccine and then you end up being exposed and then you have long COVID and then you get the vaccine? Do you have any evidence to show that it helps with long COVID after the fact? Or do you have to kind of be, ahead of it before it ever gets to that point?
Yeah, you have to be ahead of it before, because when the vaccine rolled out, a lot of long COVID groups were monitoring this data, and they actually found that only in a number. So they were I think the last time I looked at the data, it was about 20% or 29% who had long COVID, got the vaccine, and felt better after. And that actually supports that in a number of people, it might be because they have lingering virus that they're getting symptoms. But it might mean, and it means that there are other groups of long COVID that we're using this word to describe a big number of people. And so some of those people are getting better, but there's also some people who report getting worse after, and there are some people who the majority are reporting no differences.
Okay. One thing that I wanted to bring up as far as kind of the misinformation talk, one thing that I'm sure you've heard and a lot of other people have heard is that COVID is just like the flu. Can you tell us why that's not true, and maybe elaborate a little bit, what the difference is?
Yeah. So SARS-CoV-2 is a member of the coronaviruses, and the flu they're influenzas. They're completely different viral families. So that's number one. Number two, the thing about it is that, again, if you look at incidence rates, or you look at the people suffering, it's very clear that COVID numbers are way, way higher in terms of death. They've surpassed flu. But in general, they are completely different viruses. We also don't, with the flu in general, if you get the flu, you clear the virus, your symptoms go away. Whereas with COVID, again, we're seeing a substantial number of people who maintain these long COVID symptoms. So these are all of the reasons that tell you that they're not similar. I mean, the one benefit here is actually that the influenzas as a viral family mutate really, really fast. And coronaviruses don't. They actually mutate really slow. And it's a fun fact. It's because they have a back button, essentially. If coronaviruses mutate and they make a mistake, they are able to go back one generation. It's not completely foolproof, but it's able to reduce a lot of the mutations they make, which is why, in general, with the flu, you need an annual shot, even though it's milder, because it mutates so fast, whereas the coronaviruses do mutate slower. But again, we are seeing really high rates at the moment just because we have such uncontrolled spread. So that's how they differ.
Okay. One thing that you had mentioned earlier was the Johnson & Johnson vaccine. For me, I was actually thinking about getting that one because it was a single shot. You don't have to go back in a month and maybe risk not having the supply, especially early on when we didn't really have a lot of supply going around.
Yep.
What's the guidance for people who may have got the Johnson & Johnson vaccine where they can just get the one shot? Maybe they don't have any idea if they need a booster, if it's even safe to get a booster, and maybe if they need to mix maybe the Moderna with the Johnson & Johnson or the Pfizer just to have hybrid immunity. Can you talk a little bit about that?
Yeah, so I actually got the J&J vaccine, and so I've been monitoring the data really closely. And the J&J vaccine is still holding its ground. I mean, we have to remember that when we started trying to like look for a vaccine, we were hoping for efficacy of like 50, 60, 70%. And the fact that all of these vaccines are over 85%, including J&J, is still really impressive. And so J&J is still preventing severe death and hospitalization. We are seeing a slight dip in its efficacy, and it's unclear. Like I mean, it's not slight. We are seeing some efficacy reductions, or people that are infected with J&J get impacted more with infection. However, we still don't have sufficient data to show that they need a booster yet. I think that we're still waiting for government guidance, but that hasn't changed yet. I think we're still trying to collect data to make a decision. And there are clinical trials going on currently in other countries where they're mixing the mRNA vaccine, so Pfizer and Moderna, they're mixing with the adenovirus, so like J&J or AstraZeneca, to see if we can see better immunity. And it does seem that like getting a booster of an mRNA might be the future for people who got the J&J. It's just waiting now for, you know, the field to make a decision about it.
Okay, speaking of different types of vaccines, there is a vaccine in Cuba that recently was approved for ages 2 to 18. Why have we not kind of reached that point in the US? Is there maybe a difference in the way the vaccine is made that makes it maybe safer for kids, or were they kind of ahead of our studies as far as going ahead and starting those trials with kids?
Yeah, so it's okay. So in the US, right, the way that we decide, or like that we authorize a vaccine or approve a vaccine, is that you have the clinical trial process, right? So you start by doing preclinical work. So when, for example, the sequence of SARS-CoV-2 was made public last January, that was when scientists started running experiments to see what part of the, like in people who are recovering, what were the antibodies and T cells targeted towards in the immune system. And then they realized that it was the spike protein. So they identified the spike as the candidate for the vaccine. So then after they develop the vaccine, you go into the clinical trial process where you have phase 1 clinical trial, which is when you take a big group of healthy people. You give them different doses, increased doses of the vaccine, just to check safety. Once you kind of pick a dose, and then also you've collected safety data in a number of people to show that like you see the immune responses that you want to see, you then go into phase 2, which is where you take two groups of people, a group that gets the vaccine, a group that gets placebo. They don't know which group they're in. The person giving the vaccine doesn't know which group they're in. And then you wait a little while, and then you unblind to see who got sick and who didn't, and which group they were in. And so at that point, you're testing, not only continuing to test safety, but also whether the vaccine works without any bias, because nobody administering knew what they got. And so at that point, if you pass, you show that it's safe and that it works, then you go into phase 3, which is where you do phase 2, but in lots of different places around the country and around the world, to show that regardless of who's giving the vaccine and where they're giving it, you're still seeing the same results, or that it's working. And so the vaccine companies went through all of that in adult over 18s, right?
Right.
They got authorization and started rolling out. In general, the way that it works is that we always start with treatments or with vaccines. You always start with older people, and then you work your way backwards, because you know we protect kids. And so the next kind of group that was authorized were the 12 plus, because they ran a phase 3 where they gave kids who were 12 and older the vaccine to show that it worked and that it was safe. The reason why they skipped phase 1 and 2 is because kids over the age of 12, they have kind of like physically, they're very similar to, you know, adults in the way that they behave and in the way that they transmit. And so we know that the dose in the vaccine was so low that it would be unlikely to cause any effects. And so then it was approved for 12 and up. However, the way that the US clinical trial process works, and also the UK, or Europe in general, is that if you want to lower the dose for kids that are smaller or younger than the age of 12, you have to start back at phase 1. You can't skip phase 1 and 2. And so in the US, the companies then went all the way back and started at phase 1, and they're testing lower doses because children are physically smaller. And so now we're going through the process, and we are estimated to get that data by the fall. And so we're hoping to get authorization for children ages 2, 3 and up by Christmas, hopefully. so that they can start getting vaccinated to protect themselves. With Cuba, I know that they have a vaccine, but they haven't actually published any of the data for it online. So it's hard to know what they did in their clinical trials. And so they might have perhaps just immediately started with a younger group, or, but it's, I mean, I can't speak to it because they haven't published the data online. I think a lot of people are asking them to, so that we can see how it might weigh up and we might learn something from what they've done.
Yeah, that's one of the challenges I had when I was kind of prepping for this. I was looking for that study because I thought it was real fascinating, and maybe you don't know, but they did a study from ages 3 to 18, but whenever they actually approved it, they approved it for ages 2 to 18. And my question was, why would that 2 to 3 year gap matter or not matter? Is that something you can speak to?
So is the paper you read about the Cuban vaccine?
Yes, about that one. Yeah.
Yeah. So I wouldn't know. I mean, generally the way that it works in the US is that the dose and the age group that's approved in the clinical trial is what it's authorized for. So they won't include people 3 and up and then authorize for 2 and up. Like we need to know what happens in people between those ages. And so I'm not sure why that's the case. It may have been a typo, or I'm not sure why they would do that.
Yeah, I would definitely be curious to maybe circle back on that in the future, just to kind of touch on that, because that was pretty odd to me, and I think maybe some people would think it's just a typo, and maybe it is. But the other thing is, does that 1 year gap matter? Because you could look at the same situation, how we were talking about ages 64 and up can get the vaccine, or 65 and up. What does that year actually make a difference for? You know what I mean?
Yeah.
And
and that's the thing. Even if it doesn't make a difference, we can't say without data. Like we can't risk it. So it's important to make sure we
yeah,
know first.
that's a good point. For the Johnson specifically, there were early reports of, I think it's myocarditis, if I'm not mistaken. I do understand that a lot of people were able to get treatment to where it didn't actually become an issue, especially because they monitor, up to 30 minutes or so after the vaccine in a lot of places, just to make sure you don't have any kind of adverse effects. If I understand right, that issue was fixed in the vaccine. Do you know any more about that?
No, so the Johnson were associated with clotting.
Okay.
Right? Yeah. So So it was clotting that they were associated with, and as we've gathered more data, it's a really, really rare event. It's a very specific type of clot, and it was associated in women under the age of 50, and it caused a lot of concern. But now, as time has gone by, we've gotten better at identifying it looks responds poorly. And there's a group of people that have something called platelet factor 4 deficiency, and they know they have this disease. It's a diagnosis,
Okay.
and they're a group that respond poorly to like the J&J or the AstraZeneca vaccines, and so they were advised to get the mRNA. In terms of myocarditis, that was associated in young adolescents, so 12 and up, who were given the mRNA vaccines. And so just a couple of things here that I wanted to know. I know that everybody was like really freaking out about this. But just a few things, which is that like the heart itself has immune cells in it. So it would make sense that if you had inflammation going on in your body somewhere, that you might pick up signals in the immune cells that are surrounding the heart. But in general, myocarditis is something that is way more common than people realize, because typically you won't go into the hospital until, your chest really hurts, and then you go in and they scan for it. But we don't scan for it if people don't have like pain from it or obvious symptoms. And so it's, in general, we've always known that it's something that's actually really common with viral diseases. It's just most of the time it goes undetected. And then even with those myocarditis signals that showed up in young men, and this was data that came out of Israel, if you actually read the papers and looked into it, all of the boys bar one recovered without any treatment. So they just had the pang, they scanned them, they saw it, and then they recovered without treatment. And one actually required treatment, but there weren't any deaths that were noted from the myocarditis itself. And so that was where we are right now, I think, is that like continuing to collect data in the clinical trials. And I think that children are still opting into getting the vaccine, and parents are still opting in, just because the risk of myocarditis with COVID naturally far outweighs what you get with the vaccine. So kids that get infected with COVID see way more severe cases of myocarditis. And so that's why it's a risk benefit, like weighing it up.
Yeah, I think a lot of the adverse side effects people are worried about with the vaccine, in most cases, you actually have those same things that you're worried about, but actually worse off if you get COVID. Is that correct?
Yes, exactly. Because again, like remembering that with the virus, it has 29 proteins. And then also
Hmm.
the way that the virus infects your cells is that you breathe it in and then it binds to like these ACE2 receptors that you have in your airway. And when it binds to them, that process of binding activates pathways in the cell that can then cause disease and complicated symptoms. Whereas with the vaccine, it's just one protein and it doesn't engage ACE2. It basically, with the mRNA vaccines, what happens is that what you're sending in the message or the recipe for the spike protein itself. So not even the spike. And then that message, your cell has these 3D printers in it that are responsible for making proteins. And so that mRNA, that message, goes directly to those 3D printers that are sitting nowhere near your DNA. And the 3D printers already have all of the ingredients. They read the recipe and then they make the spike protein. And the spike protein is exhibited on the surface of the cell for your immune system to start, making those responses. And so you're not seeing that ACE2 engagement. It's a much cleaner process. I know people freaked out about mRNA because people saw it as like this new technology, but it's actually been around for decades. It's something that we've trialed in cancer previously. The first cancer patients to get mRNA technology were in 2011 and 2013. So we have long-term data from them. And in general, conceptually, it's a much safer way of delivering treatment because or delivering these proteins because you're using the cell as the factory. It's moderating it itself, and you're just sending the message, which then is read, and then it like disappears after a day or so. So it's not lingering around in your system. And so that's why like the disease, getting COVID-19 itself, can cause all these, complications because of
Yeah.
that engagement, and you don't see that with the vaccines.
Yeah, I think that's a big distinction from maybe some of the older vaccines whenever they're, mostly just starting to get into vaccine science, where you used to introduce part of the live vaccine, or maybe even part of the dead vaccine and introduce it that way. But now we're just showing something that looks like the vaccine. Is that something that you can talk to?
Yeah, because so vaccines can take they can be lots of different things. I mean, they can be live, they can be inactivated with heat, they can be inactivated in other ways. It just depends on the type of infection and how it infects the cells and how the immune system sees it. That's what makes the decision. So for example, with a lot of bacteria, it doesn't matter if they're alive or dead, your immune system will respond to them. With some organisms or microorganisms, you actually need them to be alive because they produce something that the immune system sees. And so like you can't just kill them because then the vaccine won't do anything. And so it just depends. By comparison, actually, the best way to kind of describe this is that J&J and AstraZeneca, both companies used a technology that has been around for a while for vaccines. It's the adenovirus. And basically what that is that they use kind of a vector or like they go into the cell. So you're injected with a virus that we know doesn't infect humans. We've identified it in chimpanzees a long time ago. It's unable to impact humans or make them sick. And that is used as the carrier to kind of escort the DNA for the spike protein into the cell. That goes into the nucleus, where again, because this virus can't integrate with human, it just borrows tools from the nucleus in order to make the messenger RNA, which is then released from the nucleus and then goes to the 3D printers, which are called ribosomes, and shows them this like recipe and says, make the spike protein. And so the mRNA vaccines are basically shortcutting that entire process and going immediately to the ribosomes and saying, make me the spike. And so that's why, like, again, in answer to your question, it just depends on the type of infection. There are a lot of viruses that we can't give live because they can be really dangerous, for example. And
Yeah.
so like you have to be able to like find what it is about them and then take that and harness that and put that in the vaccine instead to get those immune responses.
Yeah, just a kind of a side note. I think there was a lot of people kind of frustrated that we couldn't celebrate the scientific achievement of getting a vaccine out so fast. I think it was maybe a year, 2 years faster than it normally would have been. And we kind of maybe overlooked how amazing it was that we could get a vaccine out so quick and for so many people around the world also, because there's not just our vaccines that we developed. Russia has their own, I think it's a Sputnik vaccine.
Yep.
Cuba we talked about. There's a bunch of different people that made their own vaccines. Did you kind of feel like maybe we didn't get a chance to celebrate the scientific achievement, because of the pandemic and all the controversy around it?
Yeah, I mean, definitely, it definitely was incredible. But I think that the hard thing was that we were in a pandemic and things were moving like it was a specific situation. So meaning that like typically, right, if you're a company that wants to make a vaccine, you not only have to make it, but then you have to go and run these clinical trials with placebo groups that require tens of thousands of people. And then you need those people to get infected, or you need prevalent enough virus in order to reach a point where you can see whether the thing has worked or not. And obviously with SARS-CoV-2, we moved fast for a bunch of reasons. It was really easy to recruit in the clinical trials because the virus was so infectious. So the trials moved really fast. By comparison, things like Ebola, hopefully we don't have a lot of people who have it, right? For us to be able to run a trial because it's so deadly. So you don't want enough people. So that's why those trials take forever, because you're just waiting to recruit people into them. But a few other reasons that did make this situation exceptional was also that, again, everybody panicked so much because of the infectious rate. People were dying. So labs completely and labs in general pivoted their research to COVID. Two of the labs that I was in previously did COVID research alongside their own during this time. And so that meant that the science was coming out way quicker. We were, as you mentioned at the beginning when we started talking, we had a lot of preprints coming out because the science was coming out really, really fast. So that allowed us to move quickly through the preclinical trial period. And then in the clinical trials, we were able to recruit people really fast. And then the other thing that people I think forget is that these things are expensive. And so typically, if you're making a vaccine between phase 1 and phase 2, for example, you'll have to go and make money, right? You'll have to go and fundraise, and you'll go to people and you'll say, look, my vaccine has now been shown to be safe. Give me money so I can go into phase 2. And then after phase 2, you do the same to go into phase 3. No one's going to give you the money up front to do all of them. Whereas that entire part of it was eliminated, which can take years because we had public and private funding because of the pandemic. Another reason that's kind of funny is that the FDA has to sit down and meet with every single company before it moves through each of these phases. So there are only so many hours in a day, right? So typically, if you're making a treatment or a drug, it can take forever to get those in-person meetings. And I kid you not, the FDA still sends faxes. I don't know if that's changed in the last few months, but they still send faxes, which just shows you how slow and bureaucratic it can be. But again, all of that was eliminated. And so we weren't waiting around for faxes, right? The meetings were happening quicker. The trials moved faster. And so there were reasons why we managed to move so quickly, and no corners were cut here. And I think just the last thing that I'll mention that I know that you didn't ask about, but a couple of people wonder, is that why we shouldn't be worried about long-term effects, given that we've moved so fast compared to other vaccines. And I think that the key thing here is that vaccines are not like drugs or treatments. Drugs and treatments, you take them repeatedly, right, over time. So you need to monitor people to see what happens when they get a buildup in their liver or in different organs. Vaccines are like a flash or a snap, where you're showing the immune system something once or twice, so that it can go and do its thing that it does all day, every day anyway, right? It's used to generating memory responses. It just needs to see the thing. And so you're not changing anything natural about your responses. You're just showing the immune system something so it can go generate them. And so, the reason why we're not theoretically worried about long-term effects is that vaccines, you take them once or twice, that's it. They don't stay in your system for longer than a few days. And then what you're left with is your immune responses and your immune cells long term. And also, just historically, we've never seen a serious adverse event present from a vaccine longer than 6 weeks after the shot. So if you're going to have an allergic reaction, you're going to have it immediately. That's why you're asked to stay at the doctor's office for 15 to 30 minutes after, so that if you have an allergic reaction, that's your innate immune cells. It's immediate. And then you'll have a response to it, and they can give you epinephrine and treat you. And if you're going to have something that's a little bit things the clotting, or for example, there's a disease called idiopathic thrombocytopenia, ITP, that has been associated in a very small number of people, or Guillain-Barré. These are things that appear as you start to make antibodies around day 5 up to week 3. So it's just not normal. It's not biologically feasible to think that a year from now, randomly, the vaccine will have an effect on you. It's and the FDA at this point, for what it's worth, we have the first people who got the vaccine were in phase 1 trials were in March of last year, and we now have more than a year and a half worth of data from them, and we're not seeing anything unexpected. And in terms of the mRNA technology, we have data from cancer patients going back to 2011, 2013, and we're not seeing anything unexpected. And so that's why I know that these are very valid concerns people have, but these are the reasons why we're not really that concerned about it as a whole.
And just to kind of follow up on that, you said, if it's not something we take all the time to where we would have to be worried about it like drugs, like medicine. Um, if the virus was to become endemic, which there's talk about it being endemic and not necessarily a pandemic anymore, if it's something that maybe you need yearly, is it a fair comparison to say it's a lot like the flu shot would be, where maybe you get it every year, but there's no long-term side effects, even though it's kind of a yearly consistent thing? Is that something that would be
Well,
fair?
the thing is with the coronaviruses, their immune responses are lasting long term. You know, like way long. They're lasting long term, and so and again, given that the coronaviruses do mutate so slow, it's
Mhm.
not expected. I mean, again, this question depends on if we control the pandemic, because like vaccines are just one layer. There's also masking, distancing. You know, like
Yeah.
testing regularly. There are lots of things we can adopt, and so if we manage to rein in this pandemic quickly, then we'll reduce the chances of newer variants being able to emerge if, you know, the majority of the population is vaccinated and we adopt all these measures. But if it becomes endemic, it just means that hopefully what that will mean is that it's something that is circulating around, but everybody has some form of immunity in their system from the vaccine. So that means that you are not ending up in the hospital room. You're not ending up on a ventilator or in a hospital bed. The worst that you'll get is that it'll take you out for a couple of days. But we're still at the stage where we're like hoping and keeping fingers crossed that we can control this pandemic a little bit. We don't get there.
Okay. Um, so this question comes up a lot, and I think it's been part of the main controversy with the virus ever since it, was, first discovered. Um, does the virus origin matter, and why or why not?
Yeah, so a couple of things. I think that the viral origin does matter, right? Because it's important for us to know these things. I think it's important for us to have as much knowledge as possible about it if we're going to fight it or beat it.
Mhm.
I think that like there are some theories that are just not possible, right? And there are other theories that are more likely or more likely to be possible. And also, by the way, if we can work out the origin of it, we can prevent it from happening again. And we
Right.
can prevent it, we can prevent in future. And so it is important, but I think that like there are just so many moving parts here. Like, for example, we didn't identify the origin of HIV until 10 years after the original identification of it. Like these things take time, because what you have to do is that the most likely and we have the most amount of data that actually suggests that this vaccine, this virus did originate, did evolve naturally, right? Because
Mhm.
we don't see the backbone of it. We don't see the backbone of it in any other viruses that we know, and that it's possible that what happens is that as we engage in like more deforestation or wet markets, or like we're more involved with like what disturbing ecosystems that we shouldn't be disturbing, the more likely it is that we are going to get exposure to these things. And so currently virologists are like out sampling bats and pangolins, and they're trying to identify the closest cousins or relatives in order to see how exactly this virus evolved. And this virus did not evolve from SARS-CoV-1. It's a cousin of SARS-CoV-1, but not, you know, direct origin from it.
Yeah.
And so right now we're just still trying to work it out. And it's important that like we make sure that like we're collecting the data before we like make a decision, because at this point it could take a really long time because of the sampling and because of the analysis. And so it's just kind of waiting and seeing what the data says as it comes out. And you know, even recently I saw a paper today that identified one more very close relative, and so we're getting closer to seeing where the mutation happened that might have caused the evolution of SARS-CoV-2 and allowed it to then bind human cells. So yeah, I think in answer to your question, yes, I think it's important so that we can, you know, knowledge is only a good thing.
Mhm. Okay, thank you for tackling that question, because I know that comes up a lot, and I think you really gave a good answer for that. Um, I do want to ask a few listener questions. Does the vaccine affect immune compromised people in any different way? And maybe if you can speak to how that maybe affects those kind of people a little bit different.
Yeah, so it depends, because immunocompromise can be a really large umbrella. Like, for example, some people that have cancer, for example, or have
Mhm.
certain immune disorders, can take things called immune therapies, which is basically where you go in and you, like, for example, something that some cancer patients get is called B cell, you know, depletion therapy, where you basically administer a drug that eliminates their B cells and it prevents the growth of the tumor. But as you expect, if you're getting rid of B cells, you're going to impact how efficacious the vaccine can be if you don't have enough B cells. And so this is something that a lot of like immunologists have been monitoring, and it depends on the type of immunocompromised you have. There are some people that are on treatments that don't impact their response to the vaccine whatsoever. I think if it's something anyone's concerned about, they should speak to their doctor to make sure. But I know that one thing that has been adopted in some immunocompromised people who had weaker responses, so like some cancer patients, for example, is that they've had to give them repeat boosters and then they see an increase. So they'll give them like 3 shots, not just 2, in order to like really get their antibody levels and their T cell levels similar to that of like the general population.
Okay. And I know we talked about it a little bit earlier, but one of the questions that I had for you was, and this is from one of our listeners, are booster shots necessary? So if you have the 2 shots or even the one Johnson & Johnson one, even if they recommend getting booster shots, does that necessarily mean you have to get it? Like, is it necessary?
Yeah, so I think that currently the data does not support the need for a booster if you've had both shots and you're not in that group that we mentioned earlier, which is immunocompromised. And so, no, there's no data to support right now the need for a booster. But I think that we're still waiting to collect data on that. I mean, we have to see whether people who had the vaccine and then before they got a booster got infected with COVID, whether that worked as a sufficient booster or whether it's good for them to get spike protein. I mean, this is data that we're still collecting. It's the hardest part about all of this is that this virus has only been around since January of last year. And so a lot of these questions we can only answer by waiting to see what happens. So yeah.
Okay. I did have another question that comes up quite often. Why is testing not more of a focus, especially with asymptomatic people that might have COVID if they're vaccinated? And that goes with the breakthrough infections as well. Why is testing not more of a priority? And you know, just personal note, at my own work, one of the things that kind of has come up as a question is they're offering where if you have the vaccine, you don't have to get weekly testing. If you don't have the vaccine, you're required to get weekly testing. And you know, one of the points that always comes up is why is it not just required for everybody, especially with those asymptomatic cases?
Yeah, that's a really good question. And I actually completely agree that it should be more of a priority. There are a lot of groups and a lot of people actually that have really been fighting for this, including someone called Michael Mina, Mina, on Twitter. He is somebody who has, you know, been saying all year we need to make testing more accessible to people. I mean, I know that with the vaccine in general, it's because what we're seeing is that the transmission is still being driven by vaccinated and unvaccinated people mixing together. Even though vaccinated people, if they're exposed to enough virus, can get it and can transmit and can get sick, it's just far less likely, right? If you're taking any measure of precaution, it's far less likely that they are infecting at the same rate. And so that's why generally you can tell, you know, like with the vaccinated, it seems to correlate that if they're symptomatic, it likely means that, you know, they have something, whereas we don't have a lot of data actually at the moment because we're not tracking this data closely and we should be. But like we need to know a little bit more about what happens in that asymptomatic group that are vaccinated. But yeah, I think that testing should definitely be a priority. It's definitely one of those layers that people talk about that like, you can't just adopt one thing and hope that it'll work. We can't just vaccinate or just mask or just, you know, social distance. We have to adopt multiple of these things in order to see some real difference, especially given how much uncontrolled spread there is currently. And so, yeah, we should have better testing. And I think there are, like, for example, I know that in the UK, in France, I want to say actually DC, they give out free antigen tests to people to get them to regularly test, especially before they see somebody who's high risk or somebody, you know, that you wouldn't want to get infected. And so, yeah, I completely think it should be. And there is a lot of advocacy. There's a lot of people that yell about it a lot on Twitter and other social media.
Area, it's really hard to get rapid tests. You know, recently I felt sick probably a month ago and I was like, should I get tested? You know, we called around to a few different pharmacies in the area, the local clinic, the hospital, none of them had rapid tests. So potentially that means somebody might be sick and may not find out till, you know, 3 to 5 days sometimes. And I feel like, you know, rapid tests definitely have come a long way because they're available. But I think, you know, that's one area that I think everybody could agree would be an improvement.
Yeah, I absolutely agree. I think it's something that we really need to have cheaper and more
Mhm.
accessible tests so people can just stockpile them and like use them as much as possible to catch. I mean, I have a friend that caught an infection. She just like was about to go and see her parents and was like, let me do a quick test, and then showed
Yes.
positive, and so that prevented her. That probably prevented her from exposing them. And so yeah, I agree. I think it absolutely should be a priority.
Yeah, even if it's not 100%, accurate, if you get maybe a false positive, the worst case, it's an inconvenience. And that's kind of the same thing as a mask. The worst case, wearing it, maybe it's an inconvenience, but best case scenario, you don't pass the infection on. So I feel like that's kind of in the same wheelhouse as the masking situation.
Yep. Yep, exactly. And again, it's if you adopt multiple of these things, right? So
Mhm.
if you're, for example, if you're vaccinated, that's one layer, but you're also social distancing and not hanging out with people indoors. Or if you're hanging out with people
Yeah.
indoors, you're making sure that everything's ventilated properly. Or if or you're also wearing masks. Like if you do get exposed, even if somebody's in your house, but you've adopted a couple of these measures, it reduces your chances substantially that you'll have an infection on your hands. So it's like thinking about it in this way, I think will help, and as masks are just another layer that help protect.
Well, definitely appreciate you taking the time. I just, have one more question before we wrap up. And, before I do that, I definitely want to just personally, like we talked about all the testing that is being done. I just personally want to say a thank you to any lab technicians, because I know that that must be one of the busiest jobs right now, just processing those tests day in and day out
Yep.
for almost 2 years now. So I'll put that out there in the universe. My last question for you, uh, what would be the main takeaway that you'd want people to know, either from our talk today or from your work in general going forward?
Yeah, so I think that the main thing to remember is that there is just like we're all suffering from like
Yes.
information overload and exhaustion at this point. I mean, all of our lives have been impacted in like some people more terrible than others in the last year and a half. And so I think that the main thing is that I think the conversation now, it's clear that these vaccines do work. It's clear that I know that there's a murky history, with certain parts of
Yeah.
healthcare, and actually, I think the worst is that a lot of people have had personal, really bad experiences with doctors or healthcare workers, or just don't have a doctor that they trust. And so for them, it's hard for them to walk into this blindly. But I will tell you that, like, this is a worldwide effort. There are so many involved here. It's not just immunologists, it's virologists, like so many people have a seat at the table that are watching. All of the data is provided online. And again, like so many people are watching with different incentives and that are looking at this and before they make a decision for themselves and their family. And I think that it's clear that like the people that are in the ICU or in the hospital are people that have the virus, not people who are vaccinated, right? So it's clear here that the vaccines work. I know it's really scary, but like I think that this one thing that, Chris, I really enjoyed this conversation. I think it's a couple of points that you hit on is that at this point we've kind of moved on from the conversation of vaccine yes or no, and it's more nuanced now where we're talking about how can we get testing up, right? Like when is a booster going to be needed, if it's going to be needed, like how we're going to get access to the rest of the world, like things like this, immune cell dynamics, like these are the things that like are the question at the moment. And I think that like, yeah, like we can make a real difference if we talk about things that can like things like increasing testing and like really putting pressure on people to like put some effort into that. And yeah, so anyway, that's, I guess, my take home is that I think the conversation has moved on now, and I think it's clear that like vaccinating and giving your immune system a head start is probably the best thing in general, to protect during this time, just given that we continue to have uncontrolled spread. And so yeah, just like I know everybody's trying their best, and we've made it this far, and hopefully we can get more data that can help us get out of this situation sooner
Yeah.
rather than later.
Yeah. And again, I definitely want to thank you because, one of the big things that people I hope would see is that there's people like you that are out there really educating. And to kind of throw myself under the bus, I don't have the biggest platform. I'm not well versed in all the scientific studies or anything with viruses or immunology. But the fact that you're willing to open yourself up to questions that me and everyday folk have, I feel like that is a really positive message for everybody, just to know that there's people out there that will take the time even after 2 years, to still have that conversation and still get the message out there. So I appreciate you, and I definitely hope that you have a good dinner.
Of course. Thank you so much for having me. And yeah, it's we all have stuff that we're experts in, and it's just, as much as I can help people, get access to the right information, I'm happy to do it. So thank you so much again for having me.
You as well. And like I said, you have a good day, and thank you very
You
much.
too. Take care. Bye bye.
I wanted to take some time to thank everyone for listening and sharing and just being part of the podcast community with me on this journey. One of the things I do is try to get some really good content and guests that really specialize in certain areas that maybe myself or people that I know really are curious about, like this guest and like my last one. So with that being said, if you know anybody that you think would be interested in doing a podcast that would be a really good guest for a specific subject or a variety of subjects, let me know, reach out, and get a hold of me, and we'll talk about it and see if we can maybe make it happen. Thank you.